Today we're very excited to have Samuel Canner from the Jeffrey Gray Lab, and his talk is the proteome as a lectome, predictions from deep learning propose substantial protein carbohydrate interplay. And a little about him: he's a molecular biophysics PhD candidate at Johns Hopkins University, studying in the Rosetta Commons lab of Jeffrey J. Gray. His research focuses on studying molecular interactions of proteins with primary interest in non-covalent protein-carbohydrate interactions using deep learning. And he's set to defend his dissertation in the summer, excited to begin a career developing novel computational methods. And outside a lab, he enjoys spending time running 100-mile races in the wilderness, bouldering and kayaking in the open water. Wow, that's very cool. It's great to have these very two ways of exploring the world.
Yeah. So as I said, I'm Sam, and I'm here to talk to you about the proteome as a lectome. This, hopefully that you'll find this interesting. This is primarily focused on the protein carbohydrate interactions, primarily like cell-cell interactions and how they communicate and using deep learning to do that. So first of all, since this audience is more type 1 diabetes focused, I'm going to give a quick overview about what carbohydrates are and sugars of the sugar science. So first of all, carbohydrates, they're essentially ring-based molecules that are decorated in hydroxyls. What this means is they're essentially very water-like and are very transient and dynamic molecules, and they can be represented as these sticks or as cartoons of circles or squares with different colors. And they typically also, in the cell, they get linked together and form different 3D molecules where they can then also they get formed into more complex structures, such as these large branched trees, as shown in here in A, with N-glycans, where they're attached to proteins, or B, where they also attach to proteins with these cores with oglycanacs. But then there's also these carbohydrates also get linked to lipids, such as glycosphingolipids, as shown here in C, with several different ones shown, which are also ganglioside lipids. So with that, what in the cell it looks more like is this. These carbohydrates are ubiquitous across the entire cellular membrane. So they're critical to just pretty much all cell-cell interactions and all protein cell interactions because these carbohydrates cover the entire surface of eukaryotic cells. These things known as the glycocalyx. So several different glycans are shown, such as the N-glycans and some O-glycans, such as hyaluronan and heparan sulfate. But then we also have gangliosides. And so proteins interact with these carbohydrates from one cell to another or binding to different cells and just having different cell-cell communications. And typically this happens through proteins known as lectins. However, that's a very conserved and specific type of protein. But we also know that there are a lot of other families of proteins that also bind to carbohydrates, such as frizzled and wintless, as shown here in the middle. Sorry, frizzled, which is part of the wintless pathway, which binds to gangliosides. So there's this mismatch of like, how do these cells communicate with each other through these proteins? We don't have a full idea of how that happens. So that's my whole goal, is to identify a protein-wide atlas of protein carbohydrate interactions. I want to identify the lectome, how all proteins identify all the proteins that interact with carbohydrates. This is the
Sam,
primary.
can I just ask a quick question
Yeah.
here? I know you invited questions during the talk. Do you have any sort of knowledge of how the immune cells, with their sort of T cell receptor, B cell receptor entities, might or might not interact with this carbohydrate fringe?
So there's multiple different studies of this. Primarily, my assumption would be through the typical antigen display, where like an antibody shown here on the right would bind to a non-self glycan for bringing in the immune system, which would then bind the FC of that antibody with the FCR, or we would have TCR that would essentially take in the other ones and display it on the MHC, where that peptide would be glycosylate. That is my current assumption for that.
Yeah. That sounds right. So there's room for just sort of like interface between the flat cell membrane with these carbohydrate moieties and the immune cell receptors.
Yes,
Great.
absolutely.
Thanks. I just wanted to kind of illustrate that.
Okay. So get into focus on antibodies a little bit, but this goes to the whole goal of my thesis, which is to elucidate all carbohydrate interactions on proteomic scales. We can just kind of think of the entire human proteome, all the proteins in the humans. They're kind of this gray mixture of they might bind to carbohydrates, they might not. And my goal is to try to color them in blue or red, whether they bind to carbohydrates or whether they do not bind to carbohydrates. And this is actually very important to one of our collaborators, Dr. Ronald Schnarr at JHMI, the Johns Hopkins Medical Institute, where they actually performed this kind of experiment with gangliosides. Essentially, they're able to take a lipid with specific glycan on it, gangliosides, which are very important with the basal ganglia, the neural cells, and essentially, they're able to do cross-linking experiments where they put these lipids inside cells, zap it, and identify all the proteins that are nearby, run mass spec, find out which protein was nearby and actually interacts with this for understanding the cellular pathways. And so they developed 6 different probes and ran it on 2 different cell types, and were able to identify 873 putative proteins that specifically interact with gangliosides across those 9 experiments. So I want to essentially be able to take this, but do it in a computer instead. So this way we can help other people perform these types of experiments. So the whole goal is to identify all protein carbohydrate interactions in an organism. So to do this, we first need to have a protein carbohydrate data set to just benchmark against. And we're going to use that to train a neural network that we're calling PyCap. And then we're going to validate that against the experimental results of the Schnarr lab. So first off, we need a data set. And we need to characterize this data set by proteins that bind to carbohydrates and proteins that don't bind to carbohydrates. And so it's very easy to know what kind of proteins binds to carbohydrates and which ones don't, because there's a lot of solved structures and experimental evidence. You can just look at different structures of proteins that are soluble that bind to carbohydrates, some that are membrane bound, and a lot of them typically take this beta sandwich fold to bind to carbohydrates. However, in biology, it's easy to say that something does bind, but it's not super easy to tell something does not bind. So use our best biophysical intuition and thought of what proteins don't bind to carbohydrates theoretically, or their purpose is orthogonal to carbohydrate binding, such as cytoskeletal components like actin and myosin, transcription factors that bind to DNA, some antibodies. Please note, some antibodies. There are many that do, but primarily solved structures are of ones that do not, and also some small molecule binding proteins. So in total, we were able to get about 9,000 proteins that bind and about 21,000 that do not bind carbohydrates. And to do this sort of work, we're going to create a neural network. And for this audience, we're going to just simplify the black box into something that maps a bunch of input features into some output labels. And specifically here, we're going to use the protein sequence and the protein structure to get out whether or not the protein, the carbohydrate binding capabilities of these proteins. So we used a graph neural network specifically, and we took in the sequence of these proteins. We fed it into this transformer called ESM2. It's kind of like ChatGPT for protein sequences, but essentially it just takes that input like glycine at position 3 and is able to better identify, oh, this glycine is probably in a beta helix and it's very evolutionarily conserved, and it's probably also next to arginine 955 in this same protein with these higher space embeddings. And so we took those embeddings and we used the structure as well with an equivariant graph convolutional neural network. Essentially what the EGNN is just looks at a single residue in the middle and looks at all its neighboring residues and just passes messages in and out of it, something called a graph convolutional layer. But essentially it passes messages in and out through these residues, trying to identify the carbohydrate binding capabilities with these structures. I'm happy to go in more detail, but you can just think of it as this large mathematical tool that we're using to perform these calculations on this data set. And so we're developing these techniques to predict two different things. We have two different architectures that are very similar. We have something called capsif2, which predicts where the protein binds to carbohydrates, and PiCap, which predicts if a protein binds to carbohydrates. So first, we're just going to take a quick detour onto the carbohydrate binding residues of proteins. And we've used capsif2 to do this, where we quantify the accuracy of this method with something called DICE score, which it's also known as the F1 score if you're into statistics. So essentially, it measures how well we predict the residues of these proteins that interact with carbohydrates. And we have this region in this region in green, which is the correctly predicted region of the protein that capsif predicts. Blue is regions that capsif predicts it to be at, but it actually doesn't bind. And red is something that capsif fails to get. And so DICE is just this measurement between 0 and 1. That's pretty much just the accuracy. And this specific case is an 82% DICE coefficient. And in total, we then histogrammed this data up into a probability density diagram for our model capsif2 in blue that outcompetes our old model of capsif-V and another model of pesto carbs. On this entire test data set, we were able to predict the protein residues that bind to carbohydrates with the best accuracy. And so we compare our results with pesto carbs. There are many cases where our method slightly outperforms theirs, and there are some times where their method slightly outperforms ours. They are both two different techniques, and although ours does the best overall, there are some cases where it is worthwhile to still run pesto carbs if it is viable. So all in all, that is capsif2. We were able to predict the residues that bind to carbohydrates with decent accuracy. We will now go to PiCap, though, that we want to, though, predict whether proteins bind to carbohydrates, which has not been done before to our knowledge. So let's take a look at PiCap. So first we can look at how do proteins look at? If we look at proteins as a single dot on this simplified diagram of something called a T-SNE diagram, there's this kind of homogeneous mixture between what proteins bind to carbohydrates and which ones don't. And we want to separate that out. And we can see very easily whether or not PiCap does a good job by seeing if it's able to effectively separate that out. And that's after training, we're able to see that very nice line dividing between carbohydrate binding proteins in blue and carbohydrate non-binding proteins in red. So we're able to separate that out, but we want to look at a little bit more detail of how does it perform on smaller subsets. And so we get about a 90% accuracy overall when we look at but then we're looking at a few different smaller subsets. Ribosomal proteins, we get 100% accuracy, and cytoskeletal proteins, we get 90% accuracy. But then when we look at some lysozymes, proteins that specifically cleave, I believe it is glucose in the lysosome, we get 84%, which is still very solid. But then when we look at designed non-binders, we only get 60%, which is not much better than random. These are proteins that were designed specifically not to bind to carbohydrates, but they are essentially just taking a protein and then just mutating a few residues in that pocket to do that. So we have an ESM embedding, so it's likely getting muddled out through that embedding, those specific changes. And then antibodies as well, we get not much better than random with only 56%. In this data set, we have about 13 carbohydrate binding antibodies and 37 non-binding. And so right now, antibodies, we are not able to effectively capture the ability of whether or not they bind to carbohydrates or not, which is honestly a very important task for us, especially in our lab where we primarily focus a lot on antibody interactions. And so that is a very big case of just trying to figure out if we can develop methods to better identify whether or not antibodies bind to carbohydrates or glycoproteins or just the protein interfaces of different expressed proteins on the cell surface, where and how it does that. We have but good overall accuracy on very evolutionarily conserved proteins. So this comes back to the work done by the Schnarr lab where they identified 873 putative proteins. And can we get what's the correlation between our method and their method? And so we plotted this to see and validate how well PyCap does. And in this figure, we show on the x-axis the number of experiments that a protein specifically showed up in their work, where in 0 experiments, the protein likely does not bind to specific ganglioside, whereas in red, we show the number of proteins. So they find about 3,000 proteins that don't bind to the ganglioside, and then slowly decrease with a higher amount of accuracy that they have with these high throughput experiments. And with the number of experiments in blue, we show the fraction of proteins that are predicted to bind to a carbohydrate, not specifically gangliosides, and it increases pretty much monotonically with the number of experiments, which is absolutely amazing because this kind of also identifies that there is probably noise in both of our methods, but we have a strong correlation between the higher accuracy of their experimental results and our PyCap model. And just a few different proteins real quick. We have frizzled 1, which we actually very much analyzed in previous work, which binds to gangliosides. ATP2A, which was identified in 3 different experiments. And then PyCap labels TMEM164 as a non-binding protein, even though it was found in 3 different experiments to likely bind to carbohydrates. It's possible that it's a false negative, but this protein does deal with ferropoptosis of polyunsaturated lipids. So it could be a false positive from their experiments, or it could be a false negative from our PyCap. We need to do further experimental results to actually identify that. But then there are also proteins that were identified as non-binders that were not binding gangliosides, such as SMAD4, which is Mothers Against Docosahexdentaplegic 4. So with that, we were able to train a method to identify whether or not a protein binds to carbohydrates and whether it does not bind to carbohydrates. But that's great in that context, but we want to find out all proteins that bind to carbohydrates. And with that, we're going to use the AlphaFold 2 proteome to identify all the proteins in an organism that bind to carbohydrates. And we're extrapolating that, and we found actually about 35 to 40% of all proteins are predicted to bind to carbohydrates. We have about 39% that are identified in E. coli, 35% in human proteome, and 38% in mice, which is more than has ever been predicted before. So let's try and validate this and just look at some random cases to see is PyCap onto something, or is it spitting some nonsense out? And so we're looking at a few different proteins here and there. We can look at GLICNAC transferase 7, which GLICNAC stands for N-acetylglucosamine, and it actually catalyzes the transfer from a GLICNAC residue, a sugar, onto another sugar. So it interacts with sugars. That's an identified binding protein by PyCap. That's fantastic. We also look at Sonic Hedgehog, which is a very well known cellular differentiation factor that essentially orients the cell in differentiation from the anterior to posterior axis, and is predicted to bind to carbohydrates, which is fantastic. But then when we look at some predicted non-binders, we have E1 regulatory subunit 1, which binds to beta amyloid precursor, which probably doesn't bind to carbohydrates, but we can't definitively say that without a series of experiments. And we also have diene axonymal heavy chain 6. It's a cytoskeletal component, which is great that it is predicted to not bind because that's what we believe biophysically. And this all agrees with our biophysical intuition and can be used actually by all people for generating their own hypotheses. So PyCap was able to predict on the human proteome, but one thing I wanted to look at as well that I found really interesting was what about unannotated proteins? In E. coli, there's about 4,000 proteins that are unique, and about 10% of those are actually unannotated. We don't know what they do. And that's And it would be great to know what every single protein inside just the simplest model that we keep studying, E. coli, what they do. And we looked at those at 393 of those proteins that were unannotated by relation to the UniProt code. And we found about 35% of them are predicted to bind carbohydrates. So what we did then was just looked at a few of them. And one of them, shown here on the left, YBFC, it has this beautiful beta sandwich motif, which, if you remember earlier, that's a very, very common methodology for carbohydrate binding, and that's very well conserved across evolution. And STFP also has that beta sandwich fold at the extra at what is we presume is the extracellular region with a transmembrane anchor at the bottom, and then we which have very high confidence from PiCap 99%. However, when we look at another protein, YACH, it has only 72%, which is a lower confidence in that. And there's no predicted residues by CAPSIF2. And it could just be a simple false positive of the PiCap algorithm, or it could be a possible biological methodology. So this provides some insight for experimentalists to try to better also uncover the missing pieces of the E. coli proteome as well. So in total, we developed a method to predict the lectome of any species, where we took a protein carbohydrate data set and fed it in to train a model called PiCap and validated against experimental results, which we were then able to use to elucidate the entire AlphaFold 2 proteome, which identified about 35% of human proteins to bind to carbohydrates. So I can now hear you all clamoring in your seats. How can I use this amazing software? And so we actually released this. You can scan the QR code if you want, or check the chat, where we have a GitHub where everything is publicly available and you can run it on your own machine. However, it can be difficult to get things up and running, and we actually were able to implement an online server as well called Rosie that it's all linked in our preprint, as well as you can just Google Rosie, and then you'll be able to actually just use that immediately and instantly without having to set up anything on your own. So with that, I'd like to just say a little bit about what we're thinking with the future is, can we, like, first off, focus on antibodies? Because antibodies, we all know them, and they're fantastic tools for this therapeutics. They're very important in also type 1 diabetes and these interactions. And we just really want to actually understand these specific interactions in a very strong way to be able to, if we can identify whether or not how an antibody binds to something, we can also then hopefully also reverse engineer and make those antibodies to do that, or help also experimentalists in these sorts of endeavors, as well as we want to try to be able to be a little bit more specific than just binds carbohydrate or doesn't bind carbohydrate, but it's also difficult to truly get a full cap on a mannose residue versus a galactose versus a glucose. So we want to simplify the problem, is take the problem between carbohydrates and specific residues to being, can we figure out whether or not it's a square or a circle or a diamond, essentially a hex or a hexnac or a sialic acid. And these are the current directions that I believe that our work will continue to go to. And with that, I'd like to acknowledge my lab, specifically my two mentors on this project, my PI, Dr. Gray, and collaborator, Dr. Schnarr, as well as Sergey Liska for helping put up the Rosie server. And with that, I'll take any questions.
Great, really great work, Sam. This is a really valuable tool for many different disciplines. Let me ask, how do you guys have any collaborators right now that are working in, the immune space, in autoimmunity per se?
I'm not aware of people in the autoimmune space, but we have many collaborations with people that are developing antibodies, such as the Dr. Spangler lab at Johns Hopkins. And we also have industry collaborations with different pharmaceutical companies, like I believe it's AstraZeneca and Moderna, that we are working with as well on projects with antibody interactions.
Yeah, but it does seem like a great space for those who have interest in kind of pushing forward to reach out to you to your team, and sort of maybe just, get a little, maybe a tour through the GitHub or in the Rosie server.
Mhm.
If needed, a lot of people that already do this can probably guide themselves, but it might be, something of interest for people to start thinking about, especially those that are working in the interface between the immune system and, the beta cell or the islet cells.
Absolutely.
Yeah, all right. This is a great talk. It's a great tool, and we suspect that a lot of people will be listening to this, and wish you the best on your upcoming defense.
All right. Thank you very much.
Thank you. Bye-bye.
Bye.